What do you have less of when you go higher up? What's less of something pushing down on you? Air pressure, yeah. So if I could, now this would be hard to do unless I like put us all inside a vacuum and started like sucking out air from the room. But if I could increase the pressure, I could increase the rate of diffusion. It's a harder example in the classroom. If I somehow could increase the pressure. So pressure systems move through. If it was a day where we were in a high pressure weather system, you technically would smell that Axe body cologne faster. So have in your notes those 3 things I can do to increase the rate of diffusion. I can increase the temperature, I can increase the pressure, and I can increase the concentration. A little later in this chapter, you guys are going to design a lab. Everyone's going to use different types of cells. Everyone's going to pick one of those factors, and you're going to actually measure how much that factor affects rate. And we can, in the lab, affect pressure. We can change temperature. We can change concentration. Okay, so be thinking about how you might design that. Be thinking about what type of cell. You guys have used lots of cells this year. Elodea cells, you've used potato cells. You can use any type of cell we've used so far this year, and you're going to design completely your own experiment. Every group's going to be different. Okay, going to have to get back with your group again. We're going to switch topics here for a second and talk about the actual structure of a membrane. In your group, with your group members, I want you to draw a basic diagram of what a cell membrane looks like. There's probably 2 or 3 major structures you should be labeling. And then once you do that, I want you to talk about the major function of the membrane and see, since this is our theme, if you can relate structure to function. Okay, so go ahead with your group, give you 2 or 3 minutes to do this. Draw a basic sketch of a cell membrane, label the major structures, and then see if you can relate how structure goes with function.
There's ones that work for structure. The first one was
All right now you should have basic structure, what it looks like, and you should have picked up at least 2 major structures, 2 different organic compounds that I would find in that membrane. Hopefully you are to a discussion at this point about what those structures, what those 2 major organic compounds are doing. Why are they in there? Remember, structure relates to function. So what are those things that you've identified doing in a membrane? What are they doing for the membrane?
the transport proteins get transmitted.
Okay, let's get back together as a group. So, first question, easier question, is what are probably the two structures that you guys identified. What two organic compounds do we know are in a membrane? What do we have? Sam, you want to give me one? Phospholipids, or if you even just had lipids, that'd be great for right now, but they're called phospholipids. What other organic compound did your group remember that the membrane needs to have? Rachel? What did your group have for your second one? Proteins, good. And you could left it, you maybe had some specific types, or maybe you just said proteins. Did any group remember the third organic compound that's in there? We don't talk about them that often. There's only two left, so you have a 50-50 shot. Nobody had the third one? You want to take a guess? It is carbohydrates. So it's okay if you didn't have it in there. It doesn't have a very major role, and we're not going to actually talk about the carbohydrates that much. Okay, now we need just a general function. What does a membrane do? You could use a function, from chapter 4 for this. What do we say, what do we write down in our groups is the general function of a cell membrane? Brenna, what did your group have?
We said that it was to block out all the substances on the outside of the cell.
Everything? You never want anything in there?
Like most of it.
Okay.
And it can take materials into it.
Okay, so it somewhat controls what can go in and out of the cell.
Yeah.
Okay, I agree. And make sure you have some definition along those lines. Now, we have to relate the structures to the function. So what is the protein specifically doing? What is the lipid specifically doing? This was the hardest part. This is the part you're probably doing right at the end. What's the role of a protein? Let's start with that one. Did any group come up with, write down with the group, what a protein's doing in that membrane? Why is it there? Eric, does your group have anything for that? Okay, and I would say more than just fluids. So things in general go in and out of the cell. All right, I agree. The function of a protein is to allow things to go in and out of the cell. Now, the structure is the harder part, but the structure of these proteins, a lot of them are just like hollow tubes. So it kind of makes sense. If I want to have things moving in and out of the cell, I need some tubes where molecules can move through. So proteins are a great way for things to move in and out of the cell. Ashley, question?
Aren't there like tails at the end of the protein that like the things come onto to transport out of the cell?
There's tails on the ends of the lipids, not on the proteins, but you're close. All right, now, the proteins are the easier one with structure and function because the proteins are literally little like transport tubes that things can go through. The lipids are a little harder, and we're going to talk about the structure and function of those a little bit more when we get into the notes, okay? All right, so you notice I didn't draw a picture. I asked you guys to draw one because I knew I had some great ones on the next slide. So in general, this is what you should have had for a diagram of your cell membrane. Now, this one down here I really like as a membrane, but this is something you probably couldn't draw in your notes. This is really more what we can probably draw in our notes, something very simple and two-dimensional. The larger red molecules, these are my proteins. You can see that structure relates to function. If I need a way for things to get in and out of my cell, well, these are nice tubes. This tube has a bigger opening than this tube, so this tube obviously can let in bigger molecules than this one, but they're going to allow things to move in and out. And then I have a whole bunch of lipids, phospholipids, you guys told me. Is your open or your class? So the lipids are here. Here's a lipid. There's another one down here. Here's another lipid. These are actually similar to lipids you built in our organic compound chapter. You guys built triglycerides. They had a backbone and 3 tails. These are called phospholipids. And when we did the notes, I said it would be nice if these phospholipids were called diglycerides, because if the triglyceride you built had 3 tails, this is the exact same thing. It just has 2 tails, backbone and 2 tails. But they don't call them diglycerides. They call them phospholipids. And when we draw them, we typically draw the backbone as a little circle with 2 tails coming off. And you see that. And it's called a phospholipid bilayer because there's 2 layers of lipids, one on the top and one on the bottom. Now, when we look at this in a 3-dimensional model, down here, this is the nicer picture that we really can't draw too easily in our notes. you can see that it's really like a whole bunch of proteins floating around in a sea of lipids. You can see this, if we look at it three-dimensionally, all those little circles are all the tops of lipids, which means all in here, sandwiched in the middle, are all the tails of the lipids. And then down here, we have another layer of the lipids and the tails. And the proteins just kind of float in between. Okay, so definition, description that we can actually write down for the structure. It's a double layer of lipids with proteins that controls the passage of some molecules into and out of the cell. Now, we said that there are actually some carbohydrates. These diagrams don't even show the carbohydrates. Your homework tonight will show you, you're going to look at a little bit more in-depth picture of a membrane. The carbohydrates typically hang off the ends of the proteins, and they literally just look like these little strings hanging off. They have more to do with cell identification, so cells can tell different cells from one another, can tell if it's a virus or a bacteria coming up to it. Doesn't have a lot to do with the transport, and so that's why we're not talking about it too much here. This model is called a fluid mosaic model because none of the pieces are glued together. You have to imagine this diagram with all these pieces in motion. Okay, up on my desk I have this bottle. Do not shake this bottle because this will fall apart because it's set up very specifically. But this is to show you the fluid mosaic model. Because when you move it gently, you see that none of these pieces are actually connected or glued to one another. And it would be the same in that picture down there if I could animate it. What's missing from this model is there's no proteins floating in these lipids. So the lipids are not glued together, so very tiny molecules might be able to sneak from one side of the membrane to the other. Now, what's going to happen, and the reason why not everything can get through, and we have to add a little bit more to this story, is the lipid has very different properties. We need to write down two words, hydrophobic and hydrophilic. The tops of the lipids, the heads of the lipids, the circle part, are hydrophilic. Let me write the spelling down here. Okay. That's this part right here. Hydrophilic, that means water-loving. The inside, the tails, are hydrophobic. That means water-fearing. So this inside part does not like water. So even though none of the pieces are connected, and you think a lot of stuff could maybe sneak through those little cracks, not a lot can pass through because of just those properties. It's like oil and water. They just won't mix with each other. Because of all these properties, the hydrophobic, the hydrophilic, the fluid mosaic model, we say that the membrane is selectively permeable, that it does have some ability to select what can permeate or go through. It can control what moves in and out of the membrane. We call that selectively permeable. Okay, so just so we can kind of see that in motion, looks a lot like the diagram sitting on my desk. All of this is constantly moving. It is not glued together. You have little gaps where things might be able to get through. But because you have hydrophobic heads and hydrophilic tails, it does kind of create that barrier where really water and oil can't really pass through because water and oil can't mix. And then here, which my model was missing, are those proteins. Those proteins just kind of float around in there, allowing things to pass through. You can see they're all different sizes. And there's those carbohydrates that we briefly talked about, those carbohydrates that can help with kind of cell response. So look at your notes again, specifically what you just wrote down with your group the last time you met. Do you have the carbs, do you have the lipids, do you have the proteins, and do you have the structure and function idea written down? Okay.